The Cambrian period opens like a wide, quiet basin of time, recorded in layers of shale that once settled through warm, shallow seas. In these early oceans, the water moved gently across broad continental shelves, carrying fine mineral grains that slowly wrapped soft bodies in stillness. These sediments later formed deposits such as the Burgess Shale, where impressions of early animals remain preserved with uncommon clarity. Within these rocks appears the form now called Anomalocaris, emerging in the fossil record during the Middle Cambrian, already shaped for life in open water. The seafloor of this time was not crowded with reefs or forests, but spread as wide plains of mud and carbonate sand. Temperatures inferred from sedimentary chemistry indicate warm surface waters, consistent with the greenhouse climate of the Cambrian world. In these calm shallows, early animal communities gathered, their bodies rarely protected by heavy shells. Many organisms were soft, composed of tissues that normally vanish, yet here they rest as shadows pressed into stone. Anomalocaris is known only from these rare windows of preservation. Its remains occur alongside other Cambrian animals such as trilobites, wewaxides, and a wide range of lobopodians, all preserved by rapid burial in fine sediment. The environment captured in these deposits reflects low oxygen conditions at the seafloor, reducing decay and allowing delicate structures to remain. Through this quiet chemistry, the outline of a free-swimming animal became part of the geological record. In classification, Anomalocaris is placed within the radiodonts, a group that occupies the stem lineage leading toward arthropods. This position is based on shared anatomical traits, including segmented bodies and specialized frontal appendages, all documented in fossil material. These features do not match the hardened exoskeletons of later arthropods, but they display early versions of body organization that would later define the phylum. In this way, Anomalocaris rests not as a side branch, but as a step along a long path of anatomical change. The Cambrian seas supported a wide range of body plans, many of which did not persist beyond this period. Yet Anomalocaris shows a shift in ecological structure. Its fossils indicate a creature built for motion through water rather than life upon the sediment. The presence of swimming lobes and streamlined proportions reflects a departure from primarily bottom-dwelling forms. Within the marine ecosystem, this marks an expansion into the water column itself. Sedimentary layers from the Burgess Shale reveal that Anomalocaris shared its habitat with numerous small animals, including soft-bodied worms and arthropod relatives. The calm nature of these seas is recorded in the even lamination of mudstones, showing little disturbance by storms. These layers built up slowly, sheet after sheet, holding the memory of countless drifting days. Each fossil remains as a still moment in this prolonged settling. The fossil record demonstrates that Anomalocaris was already widespread during the Cambrian. Similar remains are known from the Chengjiang deposits of China and other sites of comparable age. These localities represent shallow marine settings across different continental margins, suggesting that this animal occupied broad regions of the early ocean. Its repeated appearance in these environments confirms its role as a stable component of Cambrian marine life. Unlike later periods dominated by reef frameworks, the Cambrian seabed was shaped by microbial mats and fine sediment. These surfaces provided limited structure, encouraging movement above the substrate rather than attachment to it. In such open settings, Anomalocaris would have passed through gentle currents, its body oriented to the flow of water across the shelf. The fossil record holds no evidence of burrowing or stationary behavior for this animal. Radiodonts such as Anomalocaris represent some of the earliest known animals adapted for sustained swimming. Their appearance coincides with the diversification of animal forms often termed the Cambrian radiation. This interval is marked by the first widespread preservation of complex body architectures, now catalogued across many fossil sites. Within this context, Anomalocaris stands as an early occupant of a newly explored ecological role. The sediments that preserve these fossils were deposited in basins where water circulation was restricted near the bottom. This allowed fine particles to accumulate without disruption, forming thin layers that sealed organisms where they settled. These conditions were episodic but repeated across the Cambrian world, leaving multiple archives of early marine life. Each slab of shale is therefore a fragment of a once quiet sea. Anomalocaris did not emerge in isolation. Its appearance is part of a broader pattern of diversification in marine animals, with segmented bodies and specialized appendages. These traits are observed in other stem arthropods of the period, forming a network of anatomical experimentation. The calm waters of the Cambrian shelf provided the setting in which these forms persisted long enough to leave fossil traces. The importance of Anomalocaris within these communities lies in its adaptation to active swimming. While many Cambrian animals remained closely associated with the seabed, this animal occupied the space above, moving through layered water where light filtered softly from the surface. This expansion into the water column altered the structure of marine ecosystems, introducing a mobile presence among drifting particles and small animals. In the preserved assemblages, Anomalocaris is never found attached to substrates or enclosed within burrows. Instead, it appears as isolated body parts or articulated segments lying flat within the shale, consistent with a free-swimming mode of life. The uniformity of its preservation across distant sites supports this interpretation, rooted entirely in physical evidence. The Cambrian Ocean recorded in these fossils was not turbulent. Geochemical studies indicate stable conditions in many of these basins, with little seasonal variation. This stability allowed ecosystems to persist over extended intervals, long enough for organisms like Anomalocaris to become established. The environment was therefore one of quiet continuity rather than constant upheaval. Through these records, the early marine world is revealed as a place of gentle expansion, where new forms arose without sudden disruption. Anomalocaris belongs to this unfolding, appearing when the architecture of animal life was still settling into recognizable patterns. Still, the body of Anomalocaris is known from exceptionally preserved Cambrian fossils that reveal a long form composed of repeated segments, each contributing to a smooth outline through water. These segments are not rigid plates, but flexible units arranged along the length of the animal, forming a continuous trunk that tapers gently toward the rear. Along both sides of this trunk extend broad lateral swimming lobes, flattened expansions of the body wall that overlap one another like layered leaves. The arrangement of these lobes is consistent in articulated specimens from sites such as the Burgess Shale and the Chengjiang biota, showing that this design was stable across its geographic range. Each lobe attaches to a single body segment, and together they form a chain of overlapping flaps that run from near the head region to the tail. Their shape is oval to triangular, with smooth edges rather than spines or rigid supports. The overlap is always in the same direction, with the posterior margin of one lobe resting above the anterior margin of the next. This configuration allows the body surface to remain continuous during movement, preventing gaps that would disrupt the flow of water along the sides. The fossil record preserves these lobes as flattened impressions, yet their repetition and symmetry indicate that they functioned as the primary locomotory surfaces. In life, they would have flexed in coordinated waves, producing undulating motion along the body length. This type of propulsion is inferred directly from the mechanical arrangement of the lobes, which mirror the wave patterns documented in other radiodonts preserved in Cambrian strata. No evidence exists for jointed walking limbs on the trunk, reinforcing that swimming was the dominant mode of movement. At the rear of the body, the segmented trunk terminates in a distinct tail fan. This structure is composed of several broad lobes arranged radially, forming a semicircular outline when preserved in dorsal or ventral view. The tail fan differs in shape from the lateral lobes, being wider and more rigid in appearance, suggesting a specialized function. Fossils show this fan to be centered at the posterior tip, providing balance and stability to the long body. The position and form of the tail fan indicate that it served to control direction rather than generate primary thrust. By adjusting its orientation relative to the body axis, Anomalocaris could have altered its trajectory through the water, maintaining steady movement in shallow Cambrian seas. This interpretation is supported by the consistency of tail fan structure across multiple specimens, all showing similar proportions and arrangement. The segmentation of the trunk is clearly visible in well-preserved material, where fine sediment has captured the boundaries between adjacent body units. These segments are equal in length through most of the body, creating a rhythmic repetition from head to tail. The segmentation pattern aligns with that seen in other stem arthropods of the Cambrian, linking Anomalocaris anatomically with the early stages of arthropod evolution. This feature is not inferred but directly observed in the fossil record. When viewed in side profile, the body appears laterally compressed, meaning it is taller than it is wide. This shape reduces resistance during forward motion and is compatible with sustained swimming above the seabed. The lateral compression is consistent across specimens, indicating that it is not a result of deformation during fossilization. This streamlined profile contrasts with the flattened forms of many contemporaneous benthic animals. The overlapping nature of the lobes creates a surface that would have behaved as a flexible fin running along the entire length of the body. As each segment moved slightly out of phase with its neighbors, a traveling wave would pass from head toward tail, pushing against the surrounding water. This interpretation relies solely on the physical arrangement of the lobes and does not invoke unverified behavior. The fossil evidence therefore provides a direct mechanical basis for reconstructing this mode of locomotion. The anterior region of the trunk is slightly narrower than the middle, widening gradually before tapering again toward the tail fan. This gentle variation in width is documented in articulated fossils and contributes to the overall hydrodynamic outline. There are no abrupt changes in diameter, no projections or protrusions that would disrupt smooth movement. The entire body appears shaped for steady passage through the Cambrian water column. Comparisons between specimens from different localities show the same pattern of lobes and tail fan, indicating that this architecture was conserved across populations. Material from Canada, China, and Australia all display the segmented trunk with lateral lobes and a posterior fan, confirming that these features are not local variations. This consistency suggests that the body plan was well established during the Middle Cambrian. The soft tissues that formed the lobes are rarely preserved in the fossil record, yet in these exceptional deposits they appear as dark carbon films outlining each segment. These films allow the boundaries and overlaps of the lobes to be measured, providing precise anatomical information. Such preservation confirms that the lobes were broad surfaces rather than narrow filaments, reinforcing their role in swimming. The length of Anomalocaris varied among individuals, with specimens ranging from small forms to bodies exceeding 0.5 meter. Regardless of size, the proportional relationships between segments, lobes, and tail fan remain constant. This indicates that the basic design was maintained throughout growth, with each part enlarging in harmony with the rest. The fossil record thus shows a stable developmental pattern, rather than radical changes through life. The connection between the lobes and the main trunk is seamless, without joints or hinges. This implies that the lobes were extensions of the body wall rather than independent appendages. Such an arrangement would have produced smooth waves along the body during motion, with no abrupt transitions between segments. This structural continuity is visible in slabs where the animal lies flattened, each lobe merging gently into the next. The body plan of Anomalocaris stands apart from the crawling forms that dominated earlier benthic communities. Its long segmented trunk, lateral lobes, and tail fan together form a coherent system for life in open water. Each part is preserved in the fossil record, allowing the entire design to be reconstructed without reliance on speculation. Through these remains, the shape of this free-swimming Cambrian animal becomes a quiet, steady outline drifting within ancient seas. The eyes of Anomalocaris are preserved in Cambrian fossil deposits with a clarity that allows their structure to be described in careful detail. These organs appear as rounded hemispheres, positioned at the front of the body, separate from the trunk segments and associated with the head region. Each eye is composed of thousands of small individual lenses, known from carbonaceous films and three-dimensional preservation in fine sediment. The lenses are tightly packed together, forming a curved surface that would have faced outward into the surrounding water. Fossils from the Emu Bay Shale and the Burgess Shale show these compound eyes detached from the rest of the body, indicating that they were lightly connected and composed of delicate tissues. The lenses themselves are preserved as distinct units, each one forming part of a larger optical field. Counts of these units show that the total number reaches into the thousands, a figure that places these eyes among the most complex known from the Cambrian period. No simpler visual organs are associated with Anomalocaris, reinforcing that vision was well developed. The hemispherical shape of the eyes is consistent across specimens from different regions. This geometry allows for a wide field of view, covering much of the surrounding environment. Such curvature is a physical feature visible in the fossil record and does not rely on behavioral inference. The curvature is smooth without flattened edges, suggesting that the eye surface was continuously rounded rather than angular. Each lens is similar in size, indicating a uniform array rather than a mixture of large and small visual units. This regularity suggests that visual input was evenly distributed across the eye surface. The lenses are arranged in close-packed rows that follow the curvature of the hemisphere, producing a pattern that is preserved in multiple fossils. This pattern is characteristic of compound eyes in early arthropod relatives. The size of the eyes relative to the body is notable. In some specimens, the diameter of a single eye is large compared with the width of the head region. This proportion is directly measurable from fossil material and indicates that a significant portion of the head was devoted to vision. There is no evidence of reduced or vestigial visual organs in Anomalocaris, showing that the eyes were a central anatomical feature. The presence of such complex eyes in the Cambrian fossil record demonstrates that advanced visual systems evolved early in animal history. These eyes appear in strata dated to the Middle Cambrian, placing them among the earliest known examples of compound vision. Their preservation provides a clear record of lens arrangement and eye shape, allowing paleontologists to compare them with those of other radiodonts. The position of the eyes at the front of the body would have oriented them toward the direction of movement. Fossils often show the eyes located anterior to the first trunk segments, confirming that they were part of the head region rather than distributed along the body. This forward placement is consistent with an animal that moved through the water column rather than remaining on the seabed. The lenses themselves are preserved as tiny circular or polygonal units, each separated by thin boundaries. These boundaries outline the edges of individual optical units and are visible as fine lines in well-preserved material. The regularity of this pattern indicates a controlled developmental process in the formation of the eye, even at this early stage of animal evolution. In some fossil specimens, the eyes are preserved in 3 dimensions, protruding slightly from the rock surface. This relief shows that the eyes were convex in life, projecting outward rather than lying flat against the body. Such projection would have allowed light from multiple directions to enter the visual field. The convex form is therefore a physical feature supported directly by fossil evidence. Anomalocaris lived in shallow marine environments where light penetrated the upper water column. Sedimentological data from Cambrian deposits indicate clear, well-lit waters over continental shelves. The large compound eyes of Anomalocaris are consistent with habitation in such illuminated settings, where visual detection of movement would have been possible. This relationship between eye structure and environment is grounded in the geological context of the fossils. Trilobites and other Cambrian arthropods also possessed compound eyes, but those of Anomalocaris are larger and composed of more lenses than many contemporaneous forms. This comparison is based on direct fossil counts and measurements, not inference. The difference in eye size suggests variation in visual capabilities among Cambrian animals. Bite marks preserved on trilobite exoskeletons have been associated with Anomalocaris based on the size and spacing of damage. These traces indicate interaction between Anomalocaris and other Cambrian animals. The presence of large complex eyes alongside evidence of feeding interactions supports the conclusion that vision played a role in locating prey in open water. This is not speculation, but a synthesis of physical evidence from multiple fossil types. The lenses in Anomalocaris eyes are arranged in a way that covers nearly the entire hemispherical surface, leaving little area without visual coverage. This dense packing is documented in fossil slabs where the eyes are exposed from the side. The absence of gaps between lenses suggests that the animal possessed a continuous field of view. There is no evidence in the fossil record for protective eye stalks or retractable mechanisms. The eyes appear as simple hemispheres attached directly to the head region. This arrangement is consistent across specimens, indicating that the eyes were permanently exposed rather than capable of withdrawal. The preservation of eye structure in Anomalocaris is rare among Cambrian animals, making these fossils especially valuable. Most soft tissues decay rapidly, yet here the fine details of visual organs are captured in stone. This preservation is limited to specific environments where rapid burial and low oxygen prevented degradation. The compound eyes of Anomalocaris are therefore not reconstructed from fragments but observed in multiple complete specimens. Each contributes to a growing picture of early visual evolution. Their presence shows that by the Middle Cambrian, animals had already developed complex sensory systems suited to life in open water. In this way, the eyes of Anomalocaris form quiet windows into ancient seas, recording how light once met living tissue in a world long settled into shale. The paired frontal appendages of Anomalocaris are preserved in articulated Cambrian fossils, with enough detail to describe their form and arrangement with precision. These structures project forward from the head region as a matched set, one on each side, forming the foremost elements of the body. Each appendage is composed of a series of repeated segments, arranged end to end, creating a long, gently tapering limb that extends toward the space in front of the mouth. The segmentation is visible as narrow bands in the fossil record, indicating flexible joints rather than rigid rods. Along the inner margin of each segment arises a spine, and together these spines form a row that runs the length of the appendage. The spines are slender, slightly curved, and oriented inward toward the midline of the body. Fossils show that the curvature of the entire appendage also bends inward, so that the paired limbs face one another in front of the head. This configuration is observed in multiple specimens from different Cambrian localities, confirming that it is a stable anatomical trait rather than a product of deformation. The spines vary slightly in size along the appendage, with those near the middle often longer than those at the base or tip. This pattern is recorded in well-preserved material, where individual spines can be measured directly. Their edges are smooth rather than serrated, and no evidence exists for mineralized reinforcement. The spines are therefore interpreted as soft tissue structures supported by the underlying cuticle, consistent with preservation as flattened carbon films. The number of segments in each appendage is consistent across most specimens, typically exceeding a dozen repeated units. Each segment bears a single prominent spine, and the spacing between segments is regular. This regularity indicates a controlled developmental process that produced symmetrical limbs on both sides of the body. Fossils that preserve both appendages in place show mirror image arrangements, reinforcing their paired nature. The inward curvature of the appendages is a key feature documented in articulated fossils. When preserved in lateral view, the limbs bend toward the mouth opening, forming a partial enclosure around the front of the head. This geometry is not inferred but directly visible in slabs where the animal lies flat. The consistent curvature across specimens supports the interpretation that the appendages were oriented in life toward the oral region. The mouth of Anomalocaris is preserved as a circular structure composed of radial plates, and the frontal appendages are positioned immediately anterior to this opening. This spatial relationship is evident in fossils that capture the head region in articulation. The appendages therefore frame the mouth rather than extending laterally or posteriorly. Their placement indicates that they played a role in the movement of food toward the oral opening. The fossil record also preserves specimens in which the appendages are detached, allowing their individual structure to be examined in isolation. In these cases, the segmentation and spine arrangement remain clear, demonstrating that the limbs were distinct anatomical units rather than extensions of the trunk lobes. The appendages are thicker and more robust than the lateral swimming lobes, showing that they served a different function. The spines along the appendages are directed inward rather than outward. This orientation would have brought anything held between the limbs toward the midline of the body. The curvature of the limbs further concentrates this inward focus, forming a space where captured material could be guided. This structural arrangement is consistent with the interpretation that the appendages were used to grasp and manipulate prey items in front of the mouth. Evidence for the types of prey captured by Anomalocaris includes fossils of trilobites bearing damage patterns consistent with the size and spacing of the spines. These marks occur on the dorsal exoskeletons of trilobites from Cambrian deposits. The correlation between spine dimensions and the spacing of these traces provides physical evidence for interaction between the appendages and soft-bodied or lightly armored animals. In addition to trilobite remains, the Cambrian fossil record includes soft-bodied organisms such as worms and small arthropods preserved in the same sediments as Anomalocaris. The morphology of the frontal appendages with their inward-facing spines is well suited to holding such organisms. The absence of crushing surfaces or mineralized jaws supports the conclusion that prey handling involved grasping rather than grinding. The appendages show no evidence of terminal claws or hooks. Instead, the spines gradually diminish in size toward the distal end, forming a gentle taper. This design would have allowed the limbs to close around prey rather than puncturing it. The consistent preservation of this taper across specimens suggests that it is an original anatomical feature. The joints between segments are preserved as slight constrictions, indicating points of flexibility. These joints would have allowed the appendages to bend in smooth arcs rather than sharp angles. The fossil record does not show any rigid locking mechanisms or stops, implying that the movement range was continuous along the length of the limb. Such flexibility would have been necessary for coordinating the two appendages in front of the mouth. Comparisons with other radiodonts show that the general pattern of paired frontal appendages is shared across the group, but the exact shape and spine arrangement in Anomalocaris is distinctive. Other Cambrian radiodonts possess appendages with different spine patterns or additional branching. The specific configuration in Anomalocaris is therefore unique among known Cambrian animals. The orientation of the appendages relative to the head is further clarified by specimens that preserve the entire anterior region in dorsal view. In these fossils, the appendages are positioned symmetrically on either side of the mouth opening, with their tips curving toward one another. This symmetrical placement confirms that they function together as a coordinated pair. There is no evidence that the frontal appendages were used for locomotion. Their structure differs entirely from the lateral swimming lobes, and they are confined to the head region rather than extending along the trunk. Their segmentation and spine arrangement are consistent with grasping rather than propulsion. This distinction is supported by the clear separation between trunk lobes and frontal limbs in articulated specimens. The feeding apparatus of Anomalocaris, composed of paired frontal appendages and a circular mouth, represents a novel anatomical solution within the Cambrian marine ecosystem. No other known Cambrian organism combines these elements in the same way. This uniqueness is documented through comparative analysis of fossil material from multiple taxa. Through these preserved appendages, the quiet actions of Anomalocaris are recorded in stone. The inward curving limbs lined with spines remain fixed in shale, holding the outline of a grasp that once took place in warm Cambrian waters. The mouth of Anomalocaris is preserved in Cambrian fossils as a circular structure positioned at the front of the head, separate from the trunk segments, and distinct from the paired frontal appendages. This structure is commonly called the oral cone, and it appears as a ring of hardened plates arranged around a central opening. The plates are irregular in size and shape, with no two being exactly alike, yet together they form a continuous circle that frames the entrance to the digestive system. Fossils from the Burgess Shale and Chengjiang deposits preserve this arrangement in flattened impressions, and in rare cases with slight three-dimensional relief. Each plate in the oral cone is rigid relative to the surrounding soft tissues, indicating that the mouth opening was supported by cuticular elements rather than muscle alone. The number of plates varies slightly among specimens, but the circular pattern is always present. Some plates are larger and more prominent, while others are smaller and fit into the spaces between them. This irregularity is a consistent feature of the structure and is directly observed in the fossil record. The plates do not form opposing jaws. Instead, they surround the opening evenly, creating a radial symmetry unlike that seen in later arthropods. No fossil evidence exists for hinged jaw elements, or for any division of the mouth into upper and lower components. The oral cone therefore represents a feeding structure that differs fundamentally from those of later lineages. In articulated specimens, the oral cone is located immediately behind the paired frontal appendages. The appendages curve inward toward the mouth, and their tips often overlap the outer margin of the cone in preserved material. This spatial relationship shows that the appendages delivered captured material directly to the mouth opening. The fossil record documents this arrangement repeatedly, confirming that it was not an accidental posture at burial. The plates of the oral cone are preserved as dark carbon films or as mineralized outlines, depending on the mode of preservation. Their margins are smooth, with no serrations or cutting edges. This indicates that the plates were not designed to slice or shear, but instead to frame and possibly constrain material entering the mouth. Their hardened nature would have allowed them to maintain the shape of the opening during feeding. Some fossils show the oral cone slightly collapsed or distorted, reflecting the flexibility of the surrounding tissues after death. Despite this, the radial pattern of plates remains recognizable, and the circular organization can be reconstructed with confidence. The persistence of this pattern across many specimens confirms that it is an original anatomical feature. The central opening of the oral cone is circular or slightly polygonal, reflecting the arrangement of the surrounding plates. This opening leads into the digestive tract, although the internal anatomy is rarely preserved beyond the mouth itself. No evidence exists for teeth or grinding surfaces inside the cone. Instead, the structure appears suited to guiding food inward, rather than mechanically breaking it apart. Comparisons with other radiodont fossils show that variations of this oral cone occur across the group, but the general design of a radial arrangement of plates is shared. This confirms that the structure is characteristic of Cambrian radiodonts, and not unique to Anomalocaris alone. However, the specific number and proportions of plates differ among taxa, making the Anomalocaris cone identifiable in the fossil record. The absence of true jaws is a defining feature of this feeding apparatus. Later arthropods evolved paired mandibles or other jaw elements, but Anomalocaris retained this radial system. This difference marks an early stage in the evolution of arthropod feeding structures, documented directly by the fossil record rather than inferred. In some specimens, the oral cone is preserved detached from the rest of the body, lying as a ring of plates on the shale surface. These isolated cones allow the plate arrangement to be studied without interference from surrounding tissues. Their consistent geometry across localities provides strong evidence that the structure was stable across populations. The plates themselves vary in thickness, with some appearing more robust than others. This variation is preserved as differences in relief or carbon density in fossil slabs. The irregular pattern suggests that the cone did not function as a uniform crushing surface, but rather as a flexible frame capable of slight adjustments as material passed through. The orientation of the plates is inward, with their edges facing the central opening. This is visible in fossils where the cone is preserved in slight profile. The inward-facing edges would have guided prey items toward the digestive tract. This interpretation is grounded in the observed orientation of the plates, rather than in speculation about behavior. The oral cone does not show signs of wear patterns or abrasion that would indicate heavy processing of mineralized shells. Instead, its smooth edges are consistent with interaction primarily with soft-bodied organisms. This is supported by the broader context of Cambrian ecosystems, where many animals lacked heavy armor and were preserved as soft tissues in fine sediments. In radiodont fossils, the oral cone is always associated with paired frontal appendages, reinforcing that these elements function together as a feeding system. The appendages captured and positioned prey, and the oral cone received it. This integrated arrangement is documented repeatedly in articulated material. The radial symmetry of the oral cone contrasts with the bilateral symmetry of the rest of the body. This difference is clear in fossils that preserve both the head and trunk. The cone sits at the center of the anterior region, forming a circular boundary between the external environment and the internal digestive system. No evidence exists for muscular jaws or teeth within the oral cone. The structure therefore represents a feeding mechanism distinct from that of later arthropods. This distinction is not a matter of interpretation, but a direct consequence of the preserved anatomy. Through this circular mouth, Anomalocaris interacted with its surroundings in a way now recorded only in stone. The ring of hardened plates remains fixed in Cambrian shale, holding the quiet outline of a feeding structure that has no modern counterpart, yet is known in detail through fossil preservation. Anomalocaris is known from marine sedimentary rocks that formed along shallow continental shelves during the Cambrian period, where warm seas spread across broad regions of the continents. These deposits include the Burgess Shale of what is now Western Canada and the Chengjiang biota of what is now southern China, both representing low-energy marine environments that accumulated fine mud in quiet water. The grain size of these sediments is uniformly small, showing that strong waves or currents were rare at the seafloor, and that burial of organisms occurred gently rather than through violent disturbance. The fossils preserved in these settings include abundant trilobites, small arthropod relatives, and many soft-bodied animals whose tissues would normally decay rapidly. Their preservation indicates that the seafloor lay beneath waters that limited oxygen penetration into the sediment, slowing decomposition and allowing delicate structures to be recorded. Anomalocaris occurs within these assemblages as isolated appendages, eyes, oral cones, and in rare cases partially articulated bodies, confirming that it inhabited the same environments as these other Cambrian organisms. The sedimentary layers of the Burgess Shale show repeated sequences of fine-grained mudstones, often with little evidence of bioturbation. This suggests that the bottom waters were calm enough to allow thin laminations to remain intact. In such conditions, Anomalocaris would have moved above the sediment surface rather than within it, passing through a water column that remained clear enough for light to penetrate from above. The fossils do not show abrasion or rounding, further supporting deposition in tranquil settings. Chengjiang deposits preserve a similar picture, with laminated mudstones formed in shallow marine basins near the margins of the Cambrian continent. These rocks contain a diverse community of animals, including many soft-bodied forms that occur alongside Anomalocaris remains. The consistency of preservation between these distant localities shows that Anomalocaris occupied comparable habitats across wide geographic areas. In all known occurrences, Anomalocaris is found in strata interpreted as shelf environments rather than slope or abyssal settings. The sedimentological characteristics of these deposits, including fine lamination, shallow water fauna, and absence of features associated with deep marine environments, support this interpretation. There are no fossils of Anomalocaris in rocks that formed in deep ocean basins, and no trace of this animal in turbidites or other deep water sediments. The continental shelves of the Cambrian were broad and shallow due to high global sea levels. These seas flooded large portions of continental interiors, creating extensive areas of warm, sunlit marine habitat. The presence of Anomalocaris across multiple shelf deposits indicates that it occupied these open, shallow waters rather than being restricted to a single region. Within these habitats, trilobites were especially abundant, forming a major component of the benthic community. Their exoskeletons are preserved in large numbers in the same layers that contain Anomalocaris material. This co-occurrence is a direct record of shared environment, not an inferred association. Soft-bodied organisms such as worms and lobopodians are also common in these deposits, further illustrating the diversity of life present in these calm shelf seas. The absence of Anomalocaris fossils from carbonate reef structures of the Cambrian suggests that it was not closely associated with reef frameworks. Instead, it is consistently found in mudstone-dominated assemblages. This pattern indicates a preference for open shelf environments with soft substrates rather than complex hard-bottom habitats. Geochemical studies of Burgess shale-type deposits indicate low oxygen conditions at the seafloor, even though surface waters were oxygenated. This stratification created conditions favorable for the preservation of soft tissues. Anomalocaris fossils are therefore preserved primarily in these unusual windows of geological time, where environmental chemistry allowed its remains to settle undisturbed. The geographic spread of Anomalocaris fossils includes regions that were separated by vast distances during the Cambrian. Plate tectonic reconstructions show that what are now Canada, China, and Australia were located along different margins of Cambrian continents. The repeated occurrence of this animal in these settings demonstrates that it was not confined to a narrow ecological niche, but instead occupied shallow shelf seas across much of the Cambrian world. Despite this wide distribution, there is no evidence that Anomalocaris ventured into deeper ocean settings. Deep marine environments of the Cambrian are represented by different sediment types and faunal assemblages, none of which contain remains of this animal. The absence of Anomalocaris from these deposits is consistent across the fossil record and supports the conclusion that its habitat was limited to shallow water shelves. The water depths inferred for Burgess shale-type environments are generally within the photic zone, where sunlight still reached the seafloor. This is supported by the presence of organisms adapted to light, including those with well-developed eyes. Anomalocaris, with its large compound eyes, fits within this context, though its habitat is defined here only by the sediments in which it is found. The seafloor communities preserved in these deposits include many organisms that were adapted to life on or just above the sediment surface. Anomalocaris is preserved among them, indicating that it moved within the same water masses. There is no indication of specialized adaptations for pressure or darkness that would be expected of deep ocean dwellers. Across all known fossil sites, the environmental signature remains consistent. Fine-grained sediments, abundant soft-bodied fauna, and shallow marine indicators define the places where Anomalocaris is found. This uniformity provides a clear picture of the ancient settings in which this animal drifted, moving through calm Cambrian shelf seas now preserved in layers of quiet stone. The movement of Anomalocaris through Cambrian seas is recorded indirectly in the structure of its lateral body lobes, which are preserved in multiple articulated fossils from shallow shelf deposits. These lobes extend from each trunk segment and overlap in a consistent pattern along the length of the body. The overlap is always oriented so that the posterior margin of one lobe lies above the anterior margin of the next, forming a continuous surface along each side. This configuration is visible in specimens from the Burgess Shale and Chengjiang biota, where fine sediments captured the outlines of the flaps in careful detail. Each lobe is broad and flattened rather than narrow or cylindrical, indicating that it functioned as a surface interacting with surrounding water. The lobes attach directly to the sides of the trunk, with no evidence for separate walking limbs in this region. The uniform size and spacing of these structures across specimens shows that the design was stable within the species. This consistency allows the swimming method to be inferred directly from physical anatomy rather than from analogy. When these lobes are reconstructed in life position, their overlapping arrangement forms a flexible fin running along both sides of the body. As each segment could move slightly out of phase with its neighbors, a wave of motion would pass from the front toward the tail. This wave-like progression is supported by the continuous articulation seen between lobes and trunk segments. Fossil slabs preserve the lobes in gentle curves rather than sharp bends, indicating smooth movement rather than abrupt strokes. The tail fan at the posterior end of the body complements this motion by providing a surface for steering and stability. Its broader shape differs from the lateral lobes, suggesting a specialized role at the end of the undulating wave. In specimens where the tail fan is preserved, it is centered and symmetrical, aligned with the axis of the body. This alignment is consistent with controlled forward-oriented movement through water. The combined effect of the lateral lobes and tail fan is a body plan suited for sustained propulsion rather than brief bursts of speed. There is no evidence in the fossil record for rigid fins or powerful jointed limbs capable of sudden thrust. Instead, the anatomy indicates continuous, low-amplitude motion along the trunk. This type of movement is compatible with prolonged gliding through shallow marine water columns. In Cambrian benthic animals, such as trilobites and other arthropod relatives, locomotion is recorded through the presence of jointed walking limbs adapted for contact with the substrate. These limbs are preserved as rigid appendages oriented downward from the body. Anomalocaris lacks any such structures on the trunk, confirming that it did not crawl along the seafloor. The distinction between its swimming anatomy and the walking limbs of benthic organisms is clear in articulated fossils. The fine-grained sediments that preserve Anomalocaris often show little disturbance, indicating that the animal passed above the seafloor rather than interacting directly with it. There are no associated trackways or burrows attributable to this animal. Its motion therefore took place within the water column rather than across the sediment surface. This interpretation is grounded in both anatomy and sedimentary context. The lateral compression of the body, visible in side-profile fossils, further supports a swimming lifestyle. The body is taller than it is wide, forming a narrow outline when viewed from above or below. This reduces resistance as the animal moves forward, a feature documented in the fossil record by consistent proportions across specimens. Such compression is not seen in contemporaneous crawling animals, which tend to be dorsoventrally flattened. In many fossils, the lobes appear slightly curved along their length, forming gentle arcs. These arcs are aligned in series from front to back, suggesting that the body flexed in a smooth wave rather than in isolated bends. The preservation of these curves in multiple slabs indicates that they are not the result of random deformation during burial, but reflect the original anatomical shape during movement. Radiodont relatives of Anomalocaris display similar lobe arrangements, and their fossils show comparable overlapping patterns. This repetition across taxa supports the interpretation that this swimming method was characteristic of the group. The shared anatomy allows movement style to be inferred with greater confidence, based on repeated fossil evidence rather than a single specimen. The absence of heavy mineralized exoskeletons in Anomalocaris would have contributed to a lighter body, supported primarily by soft tissues. This reduced mass is consistent with sustained swimming, as documented by the flexible nature of the lobes and trunk. Fossils preserve these tissues as carbon films, outlining each segment in quiet detail. The direction of overlap between lobes is always from front to back, a pattern that facilitates the transmission of a posteriorly traveling wave. If the overlap were reversed, movement would be inefficient, yet no such reversal is seen in the fossil record. This consistent orientation indicates that the anatomy was shaped for a specific pattern of motion. The layered arrangement of lobes creates a surface that would have interacted continuously with the surrounding water. As each lobe moved slightly downward and backward, it would have displaced water in a controlled manner, generating forward thrust. This mechanism is inferred directly from the geometry of the lobes and their attachment to the trunk. Anomalocaris fossils do not show any evidence of rigid spines or structures along the lobes that would impede smooth movement. The lobe margins are rounded and even, suggesting that they flexed freely. This smoothness is preserved in multiple specimens and is therefore a reliable feature of the anatomy. The distribution of these fossils in calm shelf environments further supports a style of locomotion adapted to open water rather than turbulent conditions. The sediments in which Anomalocaris is found show little sign of high-energy events. This implies that the animal lived in settings where slow, steady movement would have been effective for navigating the water column. The gentle curvature of the body preserved in slabs often shows the entire animal forming a subtle S shape. This pattern is consistent with wave propagation along the trunk. Such shapes are repeated across multiple specimens, reinforcing the interpretation of undulatory motion. There is no fossil evidence for attachment scars or jointed bases that would indicate use of the lateral lobes for support against the substrate. Instead, the lobes are continuous extensions of the body wall. This design differentiates Anomalocaris from crawling organisms of the same period, which possess distinct ventral appendages for substrate contact. Through these anatomical details, the movement of Anomalocaris emerges as a quiet, sustained glide through layered Cambrian waters. The overlapping lobes, the gentle curves of the trunk, and the centered tail fan together form a system recorded entirely in stone, allowing its passage through ancient seas to be traced without speculation. The fossil record preserves several lines of evidence that together reveal how Anomalocaris obtained its food during the Cambrian period. Among the most informative are trilobite exoskeletons that bear damage patterns matching the size and spacing of the frontal appendage spines. These marks are found on dorsal shields of trilobites preserved in the same sedimentary layers as Anomalocaris remains, providing a direct physical association between the two. The shape of the damage, including curved impressions and paired punctures, corresponds closely to the inward-facing spines seen on the frontal appendages. In some trilobite specimens, the injuries are incomplete, with sections of the shell missing rather than crushed. This pattern is consistent with the action of grasping structures rather than rigid jaws. The margins of the damaged areas do not show the fine grinding traces that would be expected from serrated teeth, and no mineralized jaws are known from Anomalocaris. The evidence therefore indicates that trilobites were seized and manipulated rather than mechanically shredded. Additional evidence comes from rare fossils that preserve internal contents of Anomalocaris. These specimens show masses of organic material within the trunk region, interpreted as gut contents. The preserved material lacks mineralized fragments in many cases, suggesting ingestion of soft-bodied organisms. Where mineralized fragments occur, they are few and isolated, indicating that hard parts were not the primary component of the diet. The frontal appendages and the oral cone are preserved together in articulated specimens, showing their close functional relationship. The appendages curve inward toward the mouth, and their tips often overlap the margin of the oral cone. This arrangement demonstrates that captured material was guided directly into the circular mouth opening. The physical alignment of these structures is visible in multiple fossils and does not depend on interpretive reconstruction. The oral cone itself lacks opposing jaws or cutting edges. Its hardened plates surround the opening without forming a bite surface. This anatomy indicates that food was not processed through crushing or slicing, but rather passed inward after being secured by the appendages. The smooth margins of the plates support this conclusion, as they show no adaptations for heavy mechanical breakdown. The combination of grasping appendages and a radial oral cone represents a coordinated feeding system unique among known Cambrian animals. No other contemporaneous taxa preserve the same pairing of structures. This distinctiveness is documented through comparative studies of radiodont fossils and other arthropod relatives from the same deposits. Trilobite fossils bearing bite marks are preserved in calm shelf sediments, indicating that interactions between Anomalocaris and its prey occurred in open marine environments rather than on the sea floor. The absence of sediment disturbance around these damaged trilobites suggests that feeding took place in the water column, with injured individuals settling afterward to the substrate where they were buried. There is no fossil evidence that Anomalocaris consumed carrion. Gut contents do not contain mixed assemblages of decayed remains, and the damaged trilobites do not show signs of prolonged exposure prior to burial. Instead, the injuries appear fresh, indicating interaction with living animals rather than scavenging of already dead material. This conclusion is based entirely on the physical condition of the fossils. Soft-bodied organisms preserved in Cambrian deposits, such as worms and lobopodians, occur alongside Anomalocaris remains. Their delicate tissues are recorded in the same fine-grained sediments that preserve Anomalocaris gut contents. The lack of heavy mineralization in many gut content fossils is consistent with ingestion of such soft prey. These associations are direct, grounded in co-occurrence within the same sedimentary layers. The spacing of spines along the frontal appendages matches the spacing of some bite marks on trilobite exoskeletons. This correspondence has been measured in multiple studies, providing a quantitative link between predator and prey. The regularity of these marks indicates that they were produced by repeated contact with a structured feeding apparatus, rather than by random damage. In specimens where the frontal appendages are preserved in life position, their inward curvature encloses a space directly in front of the mouth. This geometry would have allowed Anomalocaris to hold prey between the appendages and bring it toward the oral cone. The fossil record shows no alternative feeding structures that would suggest a different mechanism. The environments in which these feeding interactions are recorded are shallow marine shelf settings characterized by fine mud deposition. These sediments preserve animals where they settled from the water column, not within burrows or feeding traces on the substrate. This context supports the conclusion that feeding occurred while Anomalocaris was swimming rather than while resting on the seafloor. No trace fossils or trackways are attributed to Anomalocaris, further indicating that it did not pursue prey along the sediment surface. The anatomy of its feeding structures is oriented forward, not downward, reinforcing that interaction with prey took place in open water. This is consistent across all known fossil occurrences. The absence of heavy crushing surfaces in the oral cone suggests that hard-shelled prey were not a dominant component of the diet. Where trilobite shells show damage, it is limited and localized rather than extensive fragmentation. This indicates occasional interaction with mineralized organisms rather than routine consumption of heavily armored prey. Through these converging lines of fossil evidence, the feeding behavior of Anomalocaris is recorded without speculation. Bite marks on trilobites, preserved gut contents, and the coordinated arrangement of appendages and mouth together form a coherent picture of how this Cambrian animal obtained its food, drifting through open shelf waters and interacting directly with the living community around it. Anomalocaris is placed within a group of Cambrian animals known as radiodonts, a classification grounded in multiple shared anatomical traits preserved in fossil material. Radiodonts are defined by the presence of paired frontal appendages, a segmented trunk bearing lateral lobes, and a circular oral cone composed of hardened plates. These features occur together only in this group, and are observed repeatedly in articulated specimens from Burgess Shale-type deposits. The recognition of these shared structures allows Anomalocaris to be situated within a well-defined lineage, rather than treated as an isolated form. The frontal appendages of radiodonts share a common architecture across taxa. Each consists of a series of segments bearing inward-facing spines, preserved in fine-grained sedimentary rocks from Canada, China, and Australia. In Anomalocaris, these appendages match the radiodont pattern closely, showing the same segmentation and spine orientation documented in related genera such as Herdia and Peytoia. This correspondence is not superficial but extends to the proportions and arrangement of segments along the limb. The segmented trunk of Anomalocaris is likewise characteristic of radiodont anatomy. Radiodonts possess a long body divided into repeated segments, each bearing a lateral lobe. These lobes overlap in the same directional pattern across multiple genera, forming a continuous fin-like surface along each side of the body. Fossil slabs preserve this architecture in Anomalocaris and its relatives with remarkable consistency, confirming that this was a stable feature of the group. The oral cone is another defining element linking Anomalocaris to radiodonts. This structure appears in several genera as a ring of hardened plates surrounding a central opening, without true jaws. Although the exact number and proportions of plates vary, the radial arrangement is shared across radiodont fossils. The presence of this cone in Anomalocaris places it firmly within the same anatomical framework as other members of the group. Radiodonts are recognized as part of the arthropod stem lineage, meaning they represent early branches that precede the diversification of crown group arthropods. This placement is based on a combination of features that are intermediate between earlier Lobopodian-grade organisms and later arthropods, with fully developed exoskeletons and jointed limbs. The segmented trunk, paired appendages, and early compound eyes together illustrate a transitional stage in arthropod evolution, documented directly in Cambrian fossils. Unlike crown group arthropods, radiodonts lack mineralized exoskeletons and jointed walking limbs on the trunk. Their bodies are composed primarily of soft tissues, preserved only in exceptional circumstances. This absence of hardened armor distinguishes them from trilobites and other arthropods of the same period, and marks them as stem forms rather than direct ancestors of any single modern lineage. The fossil record of radiodonts is restricted to the Cambrian period. All known occurrences of Anomalocaris and its relatives are found in strata dating from the early to middle Cambrian, with no confirmed specimens in younger rocks. This temporal confinement indicates that radiodonts were a characteristic component of Cambrian marine ecosystems, but did not persist into later geological periods. Radiodont diversity is documented through several genera, each with variations on the core body plan. Some possess broader head shields, others display differences in appendage spine patterns, yet all retain the defining combination of frontal appendages, segmented lobe trunks, and oral cones. Anomalocaris fits comfortably within this diversity, sharing the fundamental architecture while retaining species-specific proportions. The relationship between radiodonts and other early arthropod relatives is clarified through comparative fossil analysis. Lobopodians from the Cambrian show paired appendages, but lack the complex lobed swimming surfaces of radiodonts. Later arthropods exhibit jointed limbs and hardened exoskeletons, but lack the radiodont-style oral cone. Radiodonts therefore occupy a distinct morphological position that bridges these two conditions. The stem lineage position of radiodonts is supported by phylogenetic studies that analyze shared derived traits preserved in fossils. These studies consistently recover radiodonts as basal to crown group arthropods, reflecting their mixture of primitive and derived characters. Anomalocaris contributes significantly to these analyses because of the quality and abundance of its preserved remains. Radiodont fossils occur in several Lagerstätten, including the Burgess Shale, Chengjiang Biota, Emu Bay Shale, and Sirius Passet deposits. These sites represent shallow marine shelf environments from different parts of the Cambrian world. The repeated appearance of radiodonts in these deposits demonstrates that the group was widespread and ecologically significant during this interval. Anomalocaris is among the largest known radiodonts, but its anatomy remains consistent with the group pattern. Size differences among radiodonts are documented in fossil measurements, yet the basic layout of appendages, trunk lobes, and oral cone remains conserved. This conservation supports the classification of Anomalocaris as a radiodont rather than as a separate lineage. The recognition of radiodonts as a group has refined interpretations of Cambrian ecosystems. Their presence indicates that complex necotic organisms had already evolved early in animal history. Anomalocaris, through its radiodont anatomy, represents one expression of this early expansion into the water column. Through the careful study of these fossils, Anomalocaris is no longer viewed in isolation, but as part of a broader radiodont radiation that flourished briefly within Cambrian seas. Each preserved appendage, each segmented lobe, and each radial mouthplate links it to close relatives recorded in the same ancient sediments, placing it securely within a lineage that marks an early chapter in arthropod evolution. The fossil record of Anomalocaris preserves individuals of different sizes that together document growth through time within the Cambrian seas. Specimens range from small forms only a few centimeters long to bodies exceeding half a meter, all recovered from the same sedimentary units. Despite these differences in scale, the anatomical features remain consistent, including the segmented trunk, lateral swimming lobes, paired frontal appendages, compound eyes, and radial oral cone. This uniformity across size classes shows that the overall body plan was established early and maintained as the animal increased in size. Small specimens display the same number and arrangement of trunk segments as larger ones, with each segment bearing a lateral lobe of similar shape and proportion. The spacing between segments is constant, indicating that growth involved enlargement of existing units rather than the sudden addition of new ones late in development. Fossils preserve these patterns clearly, with sediment outlining each segment in specimens of all sizes. This continuity in segmentation provides direct evidence for gradual enlargement rather than abrupt transformation. The frontal appendages in smaller individuals show the same series of inward-facing spines observed in adults. Each appendage is segmented and curves toward the mouth in identical fashion regardless of specimen size. Measurements of these appendages demonstrate that spine spacing scales proportionally with overall body length. This proportional scaling supports the conclusion that appendage structure was conserved throughout growth. The compound eyes of Anomalocaris are preserved in a range of sizes. Smaller eyes possess fewer lenses but maintain the same hemispherical curvature seen in larger specimens. The relative size of the eyes compared with head width remains similar across growth stages. This indicates that visual organs developed in parallel with the rest of the body, increasing in size while retaining their fundamental organization. The oral cone shows comparable consistency. In smaller fossils, the radial plates are fewer and smaller, yet they are arranged in the same circular pattern as in larger individuals. The relationship between plate size and overall body dimensions follows a regular scaling pattern, indicating that the feeding apparatus grew proportionally rather than being replaced or reorganized. This preservation of form across size classes further supports continuous growth. Layered fossil assemblages from sites such as the Burgess Shale contain individuals of Anomalocaris of different sizes preserved together within the same sedimentary horizons. This association indicates that juveniles and larger individuals occupied the same marine habitats. There is no evidence in the fossil record for separate nursery grounds or distinct environments associated with early growth stages. The lamination of these deposits shows that the animals were buried in calm, low-energy conditions. Within these layers, small and large specimens occur side by side, their outlines captured with similar fidelity. This pattern suggests that growth took place in stable shelf environments, rather than involving migration to different habitats during development. In radiodont relatives of Anomalocaris, similar size variation is observed, with small and large specimens sharing the same diagnostic features. Comparative analysis across radiodont taxa shows that proportional growth is a common pattern within the group. This consistency across multiple genera strengthens the interpretation that Anomalocaris followed the same developmental strategy. The absence of any distinct larval forms in the fossil record is clear. All preserved specimens of Anomalocaris, regardless of size, display the full complement of adult features. There are no fossils showing radically different morphologies that would indicate a dramatic metamorphic stage. Instead, the smallest individuals already possess the defining traits of the species. This pattern of development aligns with what is observed in many Cambrian arthropod relatives, which also show gradual enlargement rather than abrupt morphological change. The fossil record captures a continuum of sizes without breaks in structural design. For Anomalocaris, this continuum is documented through numerous specimens from multiple localities. Measurements of trunk length, lobe width, and appendage dimensions across specimens reveal linear relationships between body parts and overall size. These measurements, published in peer-reviewed studies, demonstrate that growth involved uniform scaling of all major components. No body region shows disproportionate enlargement or reduction during development. The preservation of growth stages within the same sedimentary layers provides further insight into life history. It indicates that individuals remained within the same shelf environments as they matured, rather than relocating to different parts of the marine system. This conclusion is grounded in the co-occurrence of multiple size classes in identical depositional settings. The sediments that host these fossils were deposited in shallow marine basins with limited disturbance. This allowed organisms of various sizes to be buried together during episodic sedimentation events. The resulting assemblages therefore capture snapshots of the population structure of Anomalocaris at different moments in time. Growth patterns in Anomalocaris also contribute to understanding its place within radiodont evolution. The retention of the same basic anatomy throughout development suggests that the radiodont body plan was developmentally stable. This stability is a key factor in interpreting radiodonts as a coherent group within the arthropod stem lineage. Some fossils preserve small individuals with frontal appendages and eyes that are already well formed. This indicates that functional feeding and sensory systems were present early in life. The lack of evidence for non-functional juvenile stages suggests that even small individuals were capable of swimming and feeding within the same environments as larger ones. The study of growth in Anomalocaris is made possible by the exceptional preservation of Cambrian Lagerstätten. These deposits record not only adult forms but also a spectrum of developmental stages that would otherwise be lost to decay. Through this preservation, growth becomes a visible process recorded in stone. In layered assemblages where multiple generations are preserved, there is no clustering of sizes that would imply synchronized spawning events or seasonal growth patterns. Instead, sizes appear distributed across a range, indicating continuous growth over time. This distribution reflects a stable population structure within the Cambrian shelf seas. During the Cambrian period, broad shallow seas covered large portions of the continental surfaces, forming what are known as epicontinental marine environments. These settings are recorded today in fossil-bearing sedimentary deposits spread across several modern land masses. Fossils of Anomalocaris are known from sites in what are now Canada, China, Australia, and Greenland, each preserving parts of its anatomy within fine-grained marine sediments. Although these regions are widely separated today, during the Cambrian they were arranged along different margins of ancient continents, all submerged beneath warm, shallow waters. The Burgess Shale of Western Canada preserves some of the most detailed remains of Anomalocaris, including frontal appendages, compound eyes, trunk lobes, and oral cones. These fossils occur in mudstones that were deposited on a continental shelf bordering the ancient landmass of Laurentia. The sediments are fine and laminated, showing that they accumulated in low-energy marine settings. These rocks provide a clear record of shallow water ecosystems rather than deep ocean basins. In southern China, the Chengjiang biota records a similar environment from the early Cambrian. Here, Anomalocaris is represented by isolated appendages and other diagnostic elements preserved in finely laminated mudstones. These deposits formed along the margin of the South China Block, which at the time lay in equatorial latitudes. The similarity in sedimentary conditions between Chengjiang and the Burgess Shale demonstrates that Anomalocaris inhabited comparable shallow marine environments across distant continental regions. Australia provides another record of Anomalocaris in the Emu Bay Shale of South Australia. These Cambrian sediments were laid down on a continental shelf along the margin of the ancient Gondwanan landmass. The fossils from this locality include compound eyes preserved in 3 dimensions, as well as trunk fragments and frontal appendages. The preservation style again reflects calm shelf waters with fine sediment deposition. Greenland contributes additional material from the Sirius Passet Lagerstätte, where Cambrian marine sediments preserve a diverse assemblage of early animals. Anomalocaris is present in this fauna through fragments of appendages and other characteristic elements. These rocks were deposited along the northern margin of Laurentia, within a shallow epicontinental sea, similar to that recorded in Canada. Although these regions were separated by great distances, they shared a common environmental setting. All were parts of shallow continental shelves flooded by Cambrian seas. The sedimentology of each locality shows fine-grained marine deposition, with little evidence of strong wave action or deep water conditions. This consistency demonstrates that Anomalocaris was adapted to shelf environments that extended around much of the Cambrian world. Plate tectonic reconstructions show that the continents of the Cambrian were arranged differently from today, yet shallow seas fringed many of their margins. These seas were warm, as indicated by sedimentary geochemistry and the absence of glacial indicators in these deposits. The repeated occurrence of Anomalocaris in shelf sediments from multiple paleocontinents shows that it was not confined to a narrow climatic zone or a single marine province. The widespread distribution of Anomalocaris also reflects the continuity of Cambrian marine ecosystems. Trilobites, radiodonts, lobopodians, and other early animals occur together in these deposits across different regions. This pattern indicates that similar communities were established in epicontinental seas worldwide. Anomalocaris forms a consistent part of these assemblages, further confirming its broad ecological tolerance. Despite its wide geographic range, Anomalocaris is always found in marine sediments rather than in terrestrial or freshwater deposits. The rocks that yield its fossils contain marine indicators such as trilobites, echinoderm relatives, and other exclusively marine organisms. There is no evidence of Anomalocaris in non-marine environments, demonstrating that its distribution was entirely tied to the oceans. The shelf seas of the Cambrian were generally shallow, often less than a few tens of meters deep. This is inferred from the sediment types and associated faunas preserved in the deposits. Such environments allowed light to penetrate to the seafloor, supporting communities of animals with well-developed visual systems. Anomalocaris, with its large compound eyes, fits well within this ecological framework. The distribution of Anomalocaris across Laurentia, Gondwana, and the South China block indicates that it could occupy a wide range of shelf environments. It occurs in both tropical and temperate paleolatitudes, as reconstructed from plate tectonic models. This breadth suggests that the species was not restricted to a narrow environmental band, but could tolerate variations in temperature and sediment conditions within the shelf realm. In each of the regions where Anomalocaris is found, the surrounding fossil assemblages include abundant trilobites and a wide variety of soft-bodied animals. This indicates that the ecological context in which Anomalocaris lived was broadly similar across continents. The repeated association of these taxa reflects a stable pattern of Cambrian marine life distributed across the globe. The absence of Anomalocaris from deep marine sediments is equally consistent. Cambrian deep water deposits characterized by different sediment types and faunal assemblages have not yielded its remains. This absence reinforces the conclusion that its habitat was limited to shallow epicontinental seas, rather than extending into the deep ocean. Taken together, the fossil evidence shows that Anomalocaris was a cosmopolitan inhabitant of Cambrian shelf seas. Its remains are scattered across continents that were once separated by vast marine distances, yet united by similar environmental conditions. This wide distribution reflects a broad ecological tolerance grounded in the physical record of Cambrian marine geology. Anomalocaris is known only from fossils that preserve soft tissues, showing that it lacked a mineralized exoskeleton. In every well-documented specimen, the body outline appears as a thin carbon film or flattened impression rather than as rigid plates or articulated armor. The trunk segments, lateral swimming lobes, frontal appendages, eyes, and oral cone are all recorded in this delicate mode of preservation, confirming that these parts were composed primarily of flexible tissues supported by cuticle rather than by hard mineral layers. This soft construction distinguishes Anomalocaris from contemporaneous trilobites, whose calcified exoskeletons occur abundantly in Cambrian rocks of many environments. In contrast, Anomalocaris is absent from coarse sandstones, limestones, and other high-energy sediments where hard skeletons are commonly preserved. Its fossils are confined to fine-grained mudstones and shales deposited under calm conditions, indicating that only exceptional circumstances allowed its remains to survive burial and compaction. The lack of mineralized armor means that the body of Anomalocaris would not have retained its three-dimensional form after death under normal conditions. Without rigid support, tissues collapsed and decayed rapidly unless they were sealed quickly within anoxic sediment. This requirement explains why Anomalocaris is known almost exclusively from Cambrian Lagerstätten, such as the Burgess Shale, Chengjiang Biota, Emu Bay Shale, and Sirius Passet deposits, all of which share similar fine-grained preservation styles. The fossil record provides no evidence for thickened cuticle, biomineralization, or hardened plates beyond the small elements of the oral cone. Even these mouth plates are preserved as thin impressions rather than as robust skeletal parts. The remainder of the body shows smooth outlines without jointed exoskeletal margins, reinforcing the interpretation that support was provided by flexible tissues. Because Anomalocaris lacked heavy armor, its interaction with the environment depended on movement rather than physical defense. This conclusion is drawn from anatomy alone. The streamlined body shape, overlapping swimming lobes, and posterior tail fan are preserved as soft tissues, forming a locomotory system that could function only in the water column. The restriction of Anomalocaris fossils to fine-grained sedimentary environments is therefore a direct consequence of its soft-bodied nature. Where Cambrian rocks are dominated by coarse material, or show evidence of strong currents, Anomalocaris is absent. Only in settings where gentle deposition and low oxygen slowed decay does its form remain visible. These rare deposits provide the only window into its fragile body construction today preserved. Toward the close of the Cambrian period, the fossil record of Anomalocaris comes quietly to an end. In sedimentary rocks dated to the late Cambrian, remains of this radiodont become increasingly rare, and in overlying Ordovician strata they are absent altogether. This disappearance is documented across all regions where Anomalocaris is otherwise well represented, including Canada, China, Australia, and Greenland, indicating that it was not a local loss but a global pattern. The final occurrences of Anomalocaris are found in Upper Cambrian marine deposits that preserve many of the same environments seen earlier in the period. Above these layers, in sediments assigned to the Ordovician, the characteristic frontal appendages, compound eyes, trunk lobes, and oral cones of radiodonts are no longer present. This boundary marks the end of the radiodont fossil record, with no confirmed specimens extending into younger geological intervals. At the same time, Ordovician marine assemblages show an increase in arthropod groups that possess hardened exoskeletons and different feeding structures. Fossils of trilobites, crustacean relatives, and other arthropods become more diverse and abundant in these younger rocks. Their feeding anatomies include jointed limbs and mineralized mouthparts that differ fundamentally from the radiodont oral cone and frontal appendage system. These changes in the composition of marine communities are recorded in sedimentary sequences that transition smoothly from Cambrian to Ordovician without large breaks in deposition. The absence of radiodonts above this boundary therefore reflects a true biological disappearance rather than a gap in preservation. The extinction of Anomalocaris and its relatives is part of a broader reorganization of early marine ecosystems documented in the fossil record. No radiodont fossils are known from any Ordovician Lagerstätten, even those that preserve delicate soft-bodied organisms. This consistent absence across multiple continents confirms that the lineage did not persist beyond the Cambrian. The decline of Anomalocaris thus represents the close of a distinctive anatomical experiment within early arthropod evolution, recorded entirely through the stratigraphic limits of its fossil remains. Radiodont fossils provide a clear anatomical record of early stages in arthropod evolution. Anomalocaris preserves a segmented trunk, paired frontal appendages, compound eyes, and a radial oral cone, forming a combination of features that differs from both earlier Lobopodian-grade organisms and later crown group arthropods. This mixture of traits allows paleontologists to trace how arthropod body plans emerged from simpler forms documented in Cambrian sediments. The segmented body of Anomalocaris shows how repeated trunk units bearing lateral lobes predate the appearance of jointed walking limbs and hardened exoskeletons. Its frontal appendages demonstrate an early specialization of head structures for feeding before the evolution of mandibles or other arthropod mouthparts. These features, preserved together in radiodont fossils, outline transitional conditions in the stem lineage of arthropods. Although Anomalocaris and its relatives did not survive beyond the Cambrian, their anatomy remains essential for reconstructing the sequence of changes that produced later arthropods. Each preserved appendage, segment, and oral plate contributes to a framework for understanding how complex arthropod organization developed. In this way, Anomalocaris holds a foundational place in paleobiology, recording in stone an early experiment in animal design that shaped the study of arthropod origins. Anomalocaris now rests within thin layers of Cambrian shale, its body outlined in dark films that record movement without sound. These fossils lie among trilobites, Lobopodians, and other early animals preserved together in quiet marine sediments. Each slab captures a moment when fine mud settled gently through shallow shelf waters and sealed living forms beneath its weight. Through this process, entire communities became fixed within stone, forming a continuous record of early animal life. the Burgess Shale, the Chengjung Biota, the Emu Bay Shale, and the Sirius Passet deposits all preserve this same calm story. In each of these places, Anomalocaris appears as part of a broader ecosystem, never isolated from its contemporaries. Its frontal appendages, compound eyes, segmented trunk, and oral cone occur alongside the remains of organisms that shared the same waters. Together they document a complete marine setting rather than a single species in isolation. Within these laminated mudstones, the outlines of Anomalocaris often lie undisturbed, showing no signs of transport or abrasion. The preservation style indicates that the animals settled slowly to the seafloor before burial, drifting down through water that was already calm. The absence of current structures or storm deposits around these fossils reinforces this picture of stable environments. These seas were not marked by violent disturbance at the time of burial, but by steady accumulation of fine sediment. Each preserved specimen contributes to an understanding of how early Cambrian ecosystems were organized. Anomalocaris appears in association with trilobite communities that occupied the seafloor, as well as with soft-bodied organisms that drifted or crawled nearby. This repeated pattern across continents shows that similar ecological structures existed in shelf seas around the world. The fossil record does not present Anomalocaris as an anomaly, but as one participant within a consistent marine system. The motion of Anomalocaris is recorded only through the shape of its body, frozen mid-curve within shale. Lateral lobes trace the path of undulating movement, while the tail fan rests at the end of the trunk, aligned as it would have been in life. These forms do not change across layers, indicating that the anatomy was stable within its environment. The rocks preserve not action itself, but the geometry of movement captured at rest. Over time the layers built up, sealing each generation beneath the next. Growth stages of Anomalocaris are preserved together, showing individuals of different sizes within the same beds. This vertical stacking of life forms reveals that the same shallow seas supported populations through extended intervals. The environment that carried Anomalocaris remained stable long enough for its entire life history to be recorded in stone. Eventually these ecosystems vanished, not through sudden catastrophe recorded in the rocks, but through a gradual transition at the end of the Cambrian. Above the last layers that contain Anomalocaris, Ordovician sediments record different communities, and the distinctive radiodont anatomy does not return. The shale that holds its remains marks the closing of a chapter in early animal history. What remains today is not a trace of living motion, but a quiet arrangement of fossils resting in geological order. Each preserved appendage, each lens of an eye, each segment of the trunk forms part of a larger archive that extends across continents. Together these records allow the Cambrian seas to be reconstructed without speculation, relying only on the shapes left behind. Within these deposits, the early ocean is no longer an active place, but a layered memory. The fine lamination of the rocks preserves the sequence of events in calm detail, one settling after another. The stillness of these layers mirrors the stillness of the ancient waters at the moment of burial. Anomalocaris remains there, not as a predator in motion, but as an outline in shale. Its body plan now part of the geological framework of the planet. Every specimen is a point of connection between living tissue and mineral grain, showing how early animals became part of the earth itself. Through these fossils, the Cambrian marine world persists, not as sound or movement, but as a steady pattern held within stone. The shelves that once carried Anomalocaris are gone, their positions shifted by tectonic change, yet their sediments remain. In those sediments, the ancient seas continue to exist in a different form, transformed into rock, yet retaining their structure. The story ends not with disappearance, but with preservation, as the Cambrian oceans settle into quiet geological memory.